scientists have discovered that a new piece of the immune system, which could be a goldmine of potential antibiotics, has been discovered. Tests reveal that proteasome, a protein recycling structure found in every cell in our body, can produce bacteria-killing chemicals when it detects infections. The importance of this is that it offers hope amid rising resistance among microbes to drugs. Well, Professor Yivat Merbel from the Weizmann Institute of Science in Israel told us how they made this breakthrough.
So as you clearly defined it, the proteasome is the garbage can of the cells, if you will. And using a technology we developed a way back, we actually collected the ability to have dumpster diving into these garbage cans and examine which are the pieces of little proteins that are being tossed away and cleaved away like in recycling bin. And the discovery was actually to ask, what do we see there? And when we looked inside, we actually saw that a lot of these little pieces are actually antimicrobial agents. They serve as natural antibiotics that the body all the time produces, but we didn't know anything about this process before.
So, I mean, it seems incredible that we didn't know anything about it before, because, I mean, the potential for this, we all know that people are becoming resistant to antibiotics. So how can this help that in particular?
So we believe, and we showed it very nicely in the paper, but of course these are just preliminary studies because it opens a whole field of innate immunity. But you can actually, once you identify these little pieces and you can test their effect on bacteria and show that they kill different types of bacteria, even ones that are very dangerous for patients, we showed in mice that you can produce them on the outside and then provide them as therapy to mice that were infected with either bacteremia, which may cause sepsis and actually kill the mice, and we can save them and also have reduced tissue damage, and also in a model of pneumonia, which of course is a major concern. So if you can now have a whole list, and we found hundreds of thousands of such peptides, of such little pieces, you can now try to ask whether you can give them in different combinations and different patients to actually try to fight infectious diseases and perhaps other diseases that are, we have with immune compromised patients, like in the case of transplantations and cancer patients that are awaiting therapy and so on.
So you must have had a bit of a eureka moment when it happened, because the potential for this, obviously you're right at the beginning of the scientific process, but the potential for this is enormous.
Absolutely, and we had about 2 eureka moments in this, and this, I think, the best moments that a scientist can have. I was never working on microbiology, and this came out as a complete surprise. And the eureka moment was, in fact, when we looked in the proteasome in the garbage can, and we infected cells with bacteria, we actually saw that the scissors, the recycling bin, operated differently, meaning it generated a different types of these little peptides. And then we asked ourselves why that is. And we identified that if we just isolated them and tried to allow bacteria to grow in their presence, they actually died.
Okay, so what's the next step?
There are plenty of next steps, both in the basic biology of this, because the proteasome, we thought we knew almost everything there is to know about it, but obviously it opens up a completely different avenue to look in different tissues, in different disease contexts like fibrosis, autoimmunity, cancer, and so on. But from the translational perspective, and as you clearly stated in the beginning, we have to prepare for antibiotic resistance, which is becoming a really major issue health-wise worldwide. And so if we can now test these different types of pieces across different types of bacteria and understand whether we can have a therapy that will be provided to human beings with a natural source that is not so toxic, presumably. Again, we didn't prove it in humans yet. This will require clinical trials. But since the body is the generator of these products, it can actually be much safer and have less side effects than we have with the chemical antibiotics that are being treated heavily today.
And our thanks once again to Professor Yivat Merbel for that, detailing of potentially a real scientific breakthrough.